Today is the 12th Cyclodextrin Masterclass that we are holding, and on this occasion, we wanted to do something special. So we're doing two innovations at once. Uh, One is the topic itself. So today we'll talk a little bit more about science and less about commercial applications as we dive into uh, advanced drug delivery solutions with the help of the cyclodextrins. So that will be one um, innovation, and the other would be that not CarboHyde people will do the talking, but we have two bright invited young talents who will present their work and insights in this field. So first of all, and these will be two shorter lectures. So first of all, we'll hear Shima Kalantarifard from Slovakia, who will talk about cyclodextrin-based polymers in drug delivery. And then Sarah Ramos will talk about amphiphilic cyclodextrins and their many phases in drug delivery systems. So these two presentations will follow each other. Each will be about 15 minutes. And after that, we'll have the usual Q&A session. So you can save your questions or type them into the chat box during the talks. And once all the talks are over, we'll have a common discussion about all the questions that are raised. So first we'll start with Shima. And Shima is a second-year PhD student at the Polymer Institute of the Slovakian Academy of Sciences. She's working under the supervision of Abu Fazl Haidari, whom we know well, at least the ones who are familiar with the cyclodextrin polymers and their chemistry. And her thesis is entitled Supramolecular Polymeric Hydrogels for 3D Bioprinting and Injectable Therapeutics in Cartilage and Tissue Regeneration, which is a very current topic. So, Shima, if you are ready, you can start your presentation. I'll allow you the screen share, but you can turn on your microphone and camera. Okay.
You hear me?
Yes, we can hear you.
Okay. Let me check my slide.
Okay.
Okay.
We can also see the slides.
Okay, good. Well, at first, I would to say thanks to Thomas that you give this opportunity to me to present my work here. I'm a PhD student at Polymer Institute of the Slovak Academy of Sciences and working under the supervision of Dr. Haidari. Today, I would like to present our recent work on cyclodextrin epichlorohydrin cyanobromidine polymer for resveratrol delivery to enhance human chondrocyte function in cartilage repair. I will talk about 15 minutes, and if you have any questions, please write it down. We will come back to the question at the end of the session. I have divided my presentation into three main parts. At first, I will give you a brief introduction about cyclodextrin polymers and their classification. After that, I will introduce our study design and we will check the results at the end. As most of you know, cyclodextrins are cyclic oligosaccharides and they're obtained from the enzymatic hydrolysis of the starch. They are composed of glycoproteins units and they are connected and linked to each other by glycosidic bonds. Based on the number of these glycosidic bonds, we can classify them into alpha, beta, and gamma cyclodextrin, which have 6, 7, and 8 glucose units respectively. Here you can see the schematics of the beta cyclodextrin. These cyclodextrins have the same height and they are different in their cavity size. Cyclodextrins have two different type of hydroxyl group, and through this hydroxyl, it can go surface modifications and polymerizations. And also, it has a hydrophobic cavity, which is able to form complex in the presence of the hydrophobic guest. The house gas complexation is a dynamic interaction, and it's mostly governed by the force of the van der Waals interactions and other non-covalent interactions such as electrostatic, hydrogen bonding, and hydrophobic. The equilibrium constant in this kind of reaction is equal to association K divided to dissociation K, which represent here with K on and K off. I should mention that cyclodextrin has wide applications in biomedical, in cyclodextrin-based supramolecular hydrogel, cyclodextrin-based nanoparticles, and water-soluble drug cyclodextrin. And all these topics are studied in our group, but today we want to mainly focus on cyclodextrin-based nanoparticles. We can cross-link beta-cyclodextrin to make advantage from the properties of the cyclodextrin and also the advantage of the high molecular weight polymers. We can cross-link them with different cross-linkers, divalent cross-linkers like epichlorohydrin, citric acid, glutaraldehyde, or ethylenediamine, to reach beta-cyclodextrin polymer with anionic, neutral, or cationic charge. Based on the synthesis condition, for example, the ratio of the beta-cyclodextrins to cross-linker, the reaction time, and the concentration of the sodium hydroxide that we are using, we can reach beta-cyclodextrin polymer with different architects. Many research groups classified these polymers in groups of cyclodextrin capped polymer, cyclodextrin centered star polymer, polyrotexons, and so on. And we want to mainly focus on cross-linked cyclodextrin polymers. In this study, we try to cross-link beta-cyclodextrin with epichlorohydrin in the presence of the cyanoguanidine to increase water solubility, enhance absorption, and also enhance cytocompatibility of the polymers. We synthesize the polymer. At first, we start from beta-cyclodextrin in alkaline conditions, and the polymers form through polycondensation reaction. We propose two mechanisms for this interaction. As you can see here, the amine groups of the cyanoguanidine can react with the epoxide rings of the epichlorohydrin and then react with another epichlorohydrin, and this can be linked to beta-cyclodextrin molecules together, as you can see in A. The scheme B show that the amine group of cyanoguanidine and also alkoxide in beta-cyclodextrin can react with epoxide of the epichlorohydrin, and it can here we have a cyanoguanidine terminated group. We synthesize this polymer in different molar ratio of the beta-cyclodextrin to cyanoguanidine and increase the cyanoguanidine molar ratio from 0 to 10. We use cyanoguanidine in this study because it's neutral at physiological pH, while it can enhance the hydrogen bonding through guanidine moieties and dipole-dipole interaction via cyano groups. Then we try to characterize this cyclodextrin polymer cyanoguanidine with different techniques. In FCIR, the appear of the peaks related to cyano and guanidine confirm the incorporation of the cyanoguanidine group. In the next study, we measure the HNMR of these samples, but because of the overlapping of the cyanoguanidine peak with cyclodextrin, it just gives us a limited information. So we try to confirm the structure with HSQ-CNMR, and as you can see, the presence of the aliphatic carbon confirmed the incorporation of the cyanoguanidine in polymer structure. And then we suggest that the polyhydroxypropyl ether cyanoguanidine act as a cross-linker and cross-link to beta-cyclodextrin molecules together. In next, we study the phase solubility of these polymers in the presence of the resveratrol as a drug molecule. We choose resveratrol because it's a hydrophobic polyphenol with anti-inflammatory and antioxidant activity. Also, it shows chondroprotective properties. As you can see in this diagram, by incorporations of the cyanoguanidine, the slope of the phase solubility increased, which show the increase in the capacity of the solubilizing the polymer because of the increase in the hydrogen bonding interaction with drug molecules. In next, to measure the hydrodynamic size of these particles and also charge of them, we measure dynamic like scattering and zeta potential test. As you can see, for cyclodextrin polymer and its complex in the presence of the resveratrol, we see monodispersity of the particles and the size of the 4 nanometer. The size of the cyclodextrin polymer cyanoguanidine and its complex with resveratrol increase to around 18 nanometer. And zeta potentials confirm that we have particles with the just charge of slightly neutral and negative. I should mention that nanoparticles which show the zeta potential in the range of -10 to 10 consider as neutral. In next, we measure the antioxidant activity of our polymers and complex with ABTS assay. The principle of this technique is based on the scavenging ability of material to scavenge the ABTS radical cation. And we measure this study in the different concentration of the polymer. As you can see here, in the concentration of the 0.24 milligram per ml, beta-cyclodextrin show the activity around 4%, and this activity increased by cyclodextrin polymer to around 7%, and it's increased to 53% by incorporation of the cyanoguanidine. The antioxidant activity of the cyclodextrin polymer and resveratrol and also cyclodextrin cyanoguanidine resveratrol is around 90% even at lower concentration of the polymers. In next, we try to measure the metabolic activity by XII assay. This is a colorimetric method used to measure the cell metabolic activity. In this technique, uh, it's based on the ability of the metabolic active cell to reduce the XII salt to the formazone, which is in orange color. And the intensity of the is color is directly proportional to the number of the metabolic active cells. As you can see here, with increasing the cyclodextrin polymer concentration to 10 milligram per ml, the metabolic activity is decreased, which could be related to increase in cytotoxicity. In contrast, this effect is not seen in the presence of the cyanoguanidine, and I should mention that by incorporation and complexation with resveratrol, the metabolic activity is increased. In next study, we measure the polymer uptake with chondrocyte. For chondrocyte, the drug carrier with the size in the range of the 10 to 100 nanometer could be very effective. In this study, we want to measure the cellular internalization of the cyclodextrin polymer and cyclodextrin polymer cyanoguanidine. To access this, we labeling the polymers with FITC and monitor the uptake using the confocal laser scanning microscopy. As it can show in both image, 3D and 2D image, we see the successful internalization and also spatial distributions of the polymer with cytoplasm. In summary, we synthesize cyclodextrin polymers cyanoguanidine and confirm the structure with multiple techniques. These cyanoguanidine polymers enhance resveratrol loading compared to cyclodextrin polymer and also form a stable nanoscale complexation with the range size of the 4 to 18 nanometer with a slightly neutral to negative zeta potential. And also it shows excellent cytocompatibility and efficient cellular uptake and improve chondrocyte activity and extracellular matrix production. All in all, I can say that this cyclodextrin polymer cyanoguanidine-based system is promising a drug delivery platform for cartilage regeneration. At the end, I would like to say thanks to all my colleagues at Polymer Institute, a special thanks to Professor Lashik for his support and to Dr. Haydari for his support and patience, and my friend Dr. Muhaddesi Shahabenejad for her continuous encouragement, and to all projects for their financial support. And thank you all for your attention.
Thank you. Thank you very much, Shima, for the exciting presentation. It was a very nice topic. And so, as we said, we'll jump right immediately to the second topic and we'll discuss both fascinating research areas together afterwards. So, this will be pre-recorded. Of course, Sarah is here, but you'll see. Uh, I'm sorry. But you see recording just 1 second uh, till I open it. So until that, so let me introduce Sarah. Sarah Ramos is a master's student at the University of Coimbra in Portugal, at the Faculty of Pharmacy, and uh, She has a certain interest in pharmaceutical technology, in particular with cyclodextrins, and currently she's conducting a research in the Laboratory of Drug Development and Technologies as part of her uh, master's thesis, and you'll see how she uses cyclodextrins, amphiphilic cyclodextrins, in a minute. So, uh, let me know if so. You should be able to see the screen now. Uh, can you confirm in the chat box if you can see the screen with the presentation? Can you see the screen? Okay, yes, absolutely. So I'll start, and after the presentation, we'll have the discussion. So please continue typing your questions there. Okay, enjoy.
Hello everyone, my name is Sara Ramos and I'm a master's student in pharmaceutical science at the University of Coimbra, Portugal. Alongside my academic training, I'm also currently developing some research work involving amphiphilic cyclodextrins. For this reason, today I'll be presenting a topic entitled The Many Faces of Cyclodextrins. This presentation is based on the scientific article shown here, so for those who are interested, you can find some additional information in details by scanning the QR code available. After a short introduction on the fundamentals, I will focus on presenting research examples illustrating how amphiphilic cyclodextrins can be applied in drug delivery, protein delivery, and nucleic acid delivery systems. Finally, I will provide the overview of the key points discussed and address their scientific and therapeutic implications. Cyclodextrins were discovered in the late 19th century and quickly became one of the most used excipients, especially because of their unique structure and versatility. The most common applications of cyclodextrins is enhancement of solubility and bioavailability of poorly water-soluble drugs. Cyclodextrins can also improve the physical and chemical stability of multiple drugs. Cyclodextrins possess three external hydroxyl groups that serve as reactive sites for chemical modification, which has led to the development of various CD derivatives, including amphiphilic cyclodextrins that I will discuss here today. Cyclodextrins are macrocyclic structures composed of multiple glucose units linked together in the shape of a truncate cone, with a hydrophilic outer surface and a hydrophobic cavity, which can be used to encapsulate hydrophobic molecules. Depending on the number of glucose units, we can have different classifications of cyclodextrins. We can have alpha cyclodextrin with 6 units, beta cyclodextrin with 7 units, and gamma cyclodextrin with 8 glucose units. Beta cyclodextrin is the most used in the pharmaceutical industry, especially because its availability, low cost, and ideal cavity size. This happens because each type of cyclodextrin also exhibits slightly different properties. For example, while beta cyclodextrin cavity size is ideal for encapsulation, alpha cyclodextrin openings on the ring are not enough to accumulate a lot of drugs, and gamma cyclodextrin has a fairly large cavity, which is why it's unstable during complexation. However, despite the different advantages, beta cyclodextrin have a lower water solubility, which limits its applications, and that's why today we mostly use beta cyclodextrin derivatives. A special class of these derivatives are amphiphilic cyclodextrins, in which aliphatic chains are graphed on the primary or secondary faces of the CD rings. These amphiphilic cyclodextrins are so important because they have the capacity to self-assemble to form aggregates such as micelles and nanoparticles. Acylation by fatty acids is one of the earliest modifications used to prepare this type of amphiphilic cyclodextrins. However, today it is known that several factors during acylation can influence the properties of the resulting molecules, including the presence and nature of substituents on both sides of the ring, the length of the alkyl chains, the overall degree of substitution, and the type of method employed. A recent study of the self-assembling behavior of a beta-cyclodextrin revealed that, as we can see here, for a total degree of substitution below 5, there is a formation of onion-like nanoparticles or lamellar nanoparticles, while for a total degree of substitution superior to 5, a variety of shapes were observed. For example, at 6.1, we have an inverse hexagonal ultrastructure because of the substitution on the primary face of the CD. At 8.1 and above, we have a mix of these two different shapes, and at 12.4, we have lamellar nanoparticles once again. The conclusion of this study is that the morphology seems to be mainly guided by the degree of substitution and not the chain length, but the chain length appears to have an impact on reactivity, so the longer the chains are, the more they will block future substitutions. Different polymers can also be used for the formation of CD polymer amphiphils through covalent or non-covalent interactions. In covalent methods, there are two main approaches: the arm-first method, where the preformed polymer chains are grafted on the CD core, and the core-first method, that is associated with the use of living polymerization and relies on using the CD as an active core upon which polymer chains are gradually extended. We can also have the formation of amphiphilic supramolecular structures that rely on weak interactions and are prepared during the self-aggregation state. The reversible nature of these non-covalent interactions can provide systems with unique properties like stimuli-responsiveness. One example of a drug delivery system that involves the covalent linkage between beta-cyclodextrin and polymers was designed in a classical SAR shape to deliver two different anti-cancer drugs. In this design, beta-cyclodextrin was first grafted on the PMA segment that upon self-assembly forms the pH-sensitive shell, and then this structure was linked to the PLGA, which acts as a hydrophobic core of the micelles, enhancing both the stability and biodegradability of the copolymer. These star-shaped copolymers not only show a low critical micelle concentration and size, but cause severe disruption of the cell cycle, leading to high apoptosis levels. Because of the combined effect of both therapies, it was also possible to improve the therapeutic function of doxorubicin by reducing the required dose. The intracellular delivery of proteins is more and more essential for bioengineering applications. However, translocation across the cell membrane requires the introduction of cell-penetrating peptides, or CPPs. Having this in mind, in this approach, a beta-cyclodextrin derivative bearing a specific cell-penetrating peptide, the octarginine or R8, was prepared, and at the same time, the surface amino groups of different proteins were partially modified with adamantane X. These proteins were then efficiently delivered to cells through supramolecular host gas interactions between the cavity of the cyclodextrin and adamantane. This approach not only was achieved simply by mixing these two compounds without the need to perform a covalent conjugation of a protein with a cell-penetrating peptide that requires genetic manipulation which takes time and effort, but the fact that the small adamantane tags have minimal impact on a protein structure and function makes this a promising alternative for CPP-based delivery of proteins. The increased development of gene-based therapies strongly depends on efficient delivery systems capable of overcoming major limitations of nucleic acids, such as low stability or bioavailability and biodistribution, enzymatic degradation, and limited cellular uptake. Because most of these nanocarriers rely on the electrostatic interaction with negatively charged nucleic acids, cationic amphiphilic cyclodextrins represents a good alternative system. Basically, cationic amphiphilic cyclodextrins interact with the negatively charged nucleic acids, through which promote their condensation into stable structures named polyplexes. In this study, a model system of a cationic amphiphilic cyclodextrin was functionalized with two compounds, AM2 macrophage targeting peptide ligands and polyethylene glycol forming a polyplex that incorporated a small interfering RNA that was used to downregulate the colonized stabilizing factor 1 receptor, enabling the reprogramming of immunosuppressive M2 tumor-associated macrophages into a pro-inflammatory ones. This polyplex provides sufficient small interfering RNA stability, avoiding the rapid degradation observed for naked nucleic acids after systemic administration, imperfectly accumulated in tumors, resulting in a 70% reduction in tumor weight and size, thereby demonstrating targeting delivery. Simultaneously also increased the T-cell infiltration in the tumor-associated macrophage remodeling, demonstrating its significant efficiency against prostate cancer. So, in this presentation, we first discussed how cyclodextrin modification methods significantly explain the applicability of natural cyclodextrins and often allow the formation of diverse self-assembly nanomaterials. However, the fact that this science generally involves multiple steps can make production scale-up very challenging. On the other hand, cyclodextrins can not only encapsulate drugs, but also participate in interactions with biological molecules like proteins and nucleic acids via host-guest interactions, revealing their potential as universal nanocarriers. Nonetheless, a lack of detailed understanding of the structure-activity relationships and their optimization limits the insight into the full potential of this class of cyclodextrins. Lipophilic cyclodextrins, despite these limitations, hold great promise for future development, and I hope that through this presentation I have demonstrated the importance in continuing research and investment in these fields. Here's the QR code that you can access for more information about amphiphilic cyclodextrins. And all that remains is for me to thank you everyone for your attention. Thank you to our moderator, and a special thank you to my professor, Ana Rita Figueiros. I'm also available for any questions that you might have. Thank you.
So then thank you for the presentation, Sarah. So I'll invite Sarah and Shima to switch on their cameras and microphones, and we'll engage in some discussions ideally. Oh, hi, Sarah. You're still muted. Okay. Can you speak so that we can hear you? Okay. Okay, so we have seen or heard two presentations. One was about cyclodextrin polymers and their uses, and the other ones was about amphiphilic cyclodextrins that can form nanoparticles. So both topics are pretty hot, interesting, have fascinating, very distinct uses, and offer some advantages beyond what's currently known or what's currently available with the cyclodextrin. So I invite the audience to share any questions, insights about these technologies, the problems they may have, any kind of considerations, and we'll go through one by one. So we'll start with the questions. Okay, some have been maybe partially answered, but still I'll raise them. So I guess this was still for Shima. What will be the preferred dosage form? So for your study in the polymers that you studied, have you thought about that? How you would administer this formulation?
Yes, actually, it's true that in a higher concentration, we see the higher antioxidant activity, but the concentration that at end we choose to do our polymer uptake with chondrocytes and so on is the concentration of 0.125.
Okay. Uh, we also had a question if the inclusion complexes, this was still about the polymers. Are they water soluble or they are just dispersible in water? How we should envision such complexes?
They has a very high solubility.
Yes, basically that's okay, that's not necessarily true for any kind of polymer complexes, but according to most of the papers that are presented, cyclodextrin polymers have even better solubilizing capabilities than the naked or not naked the monomer cyclodextrin. So usually the same cyclodextrin polymerized can solubilize cyclodextrins at a higher extent compared to the monomer itself. And interestingly, they're often also more safe when it comes to cellular data. So it's indeed a very promising system. What methods were used to make the inclusion complexes? I think this is also for Shima. So how you make the inclusion complexes?
Actually, we put different concentration of the polymer in the presence of the higher concentration of the uh, s- resveratrol as a drug molecule, and the complexations formed by a string about 48 hours.
So technically, it's very similar process than using any other cyclodextrin. So
Yes.
it's making a common solution in an aqueous media and then uh, isolating, evaporating, I don't know, lyophilizing, whatever. So it's the same process, you mean? Yeah. So you can use the same process to make the complex. What is the rationale? Maybe this is already for Sarah. I'm not sure. So what is the rationale of using beta cyclodextrin versus gamma cyclodextrin? Okay, this is a note to self because it's hard to distinguish between the
Yeah,
questions.
I don't know where it could be the
But
question for my presentation.
since it came later, Sarah, do you have any idea why could it be more useful to use betas than gammas in your design?
Beta is mostly used because it's cavity size, as I said. Most of the drugs can be encapsulated in beta derivatives that are used um, uh, I sorry to to be a better water solubility of the cyclodextrin.
Yeah, we also have some experience at CarboHyde with creating amphiphilic cyclodextrins, and we create both beta and gamma. So here, because it's a nanoparticle formation, the drug delivery is completely different from the monomer cyclodextrin. So the cavity itself during delivery doesn't play that much of a role. But what is critical is from a synthesis perspective, the beta derivatives and the gamma derivatives are not equally complicated to make. So, depending on how we want to modify the cyclodextrin, which positions, what kind of groups we want to put in some cases we favor the beta, in some cases the gamma. So, it's in our case, at least at CarboHyde, it's not really the actual size of the cavity, but more of a practical approach, which cavity is easier to be used to create a certain derivatives. In some cases, it's the beta, in other cases, the gamma. What organic solvent do you use, and do you use low percentage of organic solvent or high concentration? So could you clarify this question about the use of the organic solvent when when do you think, or which aspect of the presentation this was relevant to? It would be easier. Elias is asking, what would be the advantage of using a CD polymer over a commercially available cyclodextrin in terms of solubility and delivery? So, Shima, this is for you.
Actually, when we have cyclodextrin polymer compared to other cyclodextrin, we can increase both solubility and also this polymer materials prepare a higher stability of the complex, and also because of the multivalency, it can increase the phase solubility of the drugs.
Yes, but I can also add because uh, I am also on the opinion that okay, cyclodextrin polymer is a new drug delivery system compared to the approved cyclodextrins, as you say here, uh, and uh, and if we think about the new excipient, it has to come with certain advantages over the existing solutions, which in my opinion has to be more than just it's a better solubilizer. What I can say is that, okay, so cyclodextrin polymers may have a different release profile for the active ingredient. So whereas the monomer cyclodextrins, like the sulfobutyl cyclodextrin you mentioned, release the active ingredient immediately. The cyclodextrin polymer is a complex system that can retain for a longer period the active ingredient, depending on how you design the polymer. So with such systems, prolonged or controlled release is easier to achieve than with the other cyclodextrins. And also, it has different physicochemical characteristics. So it can have a longer residence time in the body, higher viscosity in certain applications, like for an ophthalmic use, that can be an advantage. So I think when it comes to polymers, it's more than just the better or the higher solubility increase, but it can offer some advanced or different both physical and functional properties compared to naked cyclodextrins. Um, but yeah,
Yes, and also I can add that with preparing the polymeric cyclodextrins, we can tune this associations K and dissociation K more precisely. And with these techniques, we can also have a control on the drug release.
yes, yes. Beta cyclodextrin absorb moisture. Well, yeah, cyclodextrins are hygroscopic, so I don't know what to do about that one. You cannot do much about it. But I have a question for Sarah. So when you made this, because I think many of the attendees are familiar, probably or more familiar with making a cyclodextrin complex, but making a cyclodextrin assembly is certainly a, a more rare knowledge. So uh, how reproducible is this kind of encapsulation? Can you easily create the same encapsulation efficiency when you work with amphiphilic cyclodextrins? Is it easy to work with them to create nanoparticles? Can you tell us about this one, Sarah? Sarah, are you still here?
Yes, sorry, my camera, I don't know what's.
Okay, you can go without the
Uh,
camera.
Yeah. So my my research, we're not working on the synthesis of amphiphilic cyclodextrins. Is reported that amphiphilic cyclodextrins can be obtained by many pathways, enzymatic pathways, or different chemical reactions using different bonds. But the easiest chemical approach is to produce amphiphilic cyclodextrins in a single step is through direct esterification of native cyclodextrins. But that's allowing the substitutions to occur freely. However, if our goal is to selectively modify cyclodextrins, we have multiple steps using normally protecting groups. This is necessary due to the presence of the three distinctive types of hydroxyl groups and and yeah, hydroxyl groups of the cyclodextrin that have a different reactivities.
Yes, but I meant to ask about the encapsulation of the active ingredient. So is it easy to create a nanoparticle with these amphiphilic cyclodextrins and put a cargo into the nanoparticle? Is it a complicated process?
Uh, there is simple methods to achieve a CD drug aggregate, but the most difficult part is making sure that these aggregates have a PDI inferior to 2, making sure they have stability is sometimes difficult to evaluate how drugs can they can um, um, work inside the cyclodextrin.
Yes, I can. We have very similar experience with the GeneGut project where you said that there is no available data on the structural activity relationship, so how different amphiphilic cyclodextrins can form these nanoparticles. And okay, we haven't published, but we created like over 100 different derivatives, different cavity size modifications, and we did this structure activity studies, and it was dramatic, the differences. So some of these amphiphilic cyclodextrins were not able to form any kind of nanoparticles, and some were really excellent. And the same was true for delivering the cargo efficiently to the cells, or the encapsulation efficiency. So even though the platform seems promising, it will be very critical what kind of cyclodextrin, amphiphilic cyclodextrin, we pick at the end of the day for further development. There was a question, is there a special method to prepare samples and test the potency of the active ingredient when it's inside the cyclodextrin? Uh, I will not raise it to you. It's very general. There are a lot of techniques how you can incorporate an active ingredient into the cyclodextrin's cavity. Well, I can share with you some kind of education deck, but it's very general. But this one is for Shima. Can you explain a little about the synthesis of the CDPC and the purification?
Actually, we use the methods in the literatures, same
We have
method.
some echo. I don't know. One of the microphones should be muted, Shima.
Is it okay now?
No, but I can mute one of them. Try now.
Okay. Actually, we use the literature's reported technique to synthesize this kinds of polymers in the alkaline conditions. And in our study, the reaction time was 3 hours. At first, we put beta cyclodextrin in the solution of the sodium hydroxide with a concentration of the 33%, and after 15 minutes we add epichlorohydrin and cyanoguanidine at the same time to the mixture, and then proceed the reaction and stir it, and after 3 hours by adding acetone we end the polymerization and decantate the solution, and after freeze drying we reach the polymers and characterize them as with the technique that we talk about them. The purification technique that we use is the simple. We use a dialysis bag with the cutoff of 14,000, and doing this dialysis for 3 days with water.
Yeah, I think it's very typical that dialysis as the purification technique for the polymers. Okay. Any other questions before we conclude? Our time is soon up. But in any case, if you have specific questions related to this research or the results, or you got inspired for some collaborations, you can reach out directly to Sarah and Shima. I'm sure that they would be delighted to receive feedback from you. And in any other case, Sarah, Shima, it was excellent, and thank you for participating and uh, honoring our afternoon with your delightful talks. And I would like to thank everyone the attention, and see you around probably in 1 month at our next masterclass. So you all have a good day, a good evening, and well, yeah, take care. Thank you all for attending.
Thank you. Bye.
Thank you, ladies. Take care. Bye bye.